Okay, so hello guys and welcome back to Replication Academy. So previously we talked about the initiation, elongation and termination of the replication, right? So there are a few more important topics are there which were left. So in this lecture we'll discuss about the beta clamps or the sliding clamp which I was telling about, like, which is important as part of the replication. Okay, so the sliding clamps. Now the function and like why we are talking about these sliding clamps. So they actually increase the DNA polymerase processivity. Okay, so this is very important, like how the sliding clamps are helping in increasing the polymerase processivity. Okay, so at they act at the replication fork, and it is association of these sliding clamps with the DNA polymerase is important. Okay, so clamp means like the word itself says, उसको hold करता है, ठीक है? And they are made up of multiple identical subunits assembled in the shape of the donut. Can you see it over here? So how many subunits are there? Can anyone tell me how many subunits are there? By looking at this figure and this figure, can you tell me the number of the subunits which is required for sliding clamp?
6, ma'am.
Yes, very good. So there are 6 subunits are there. 1, 2, 3, 4, 5, and 6. And at the center you have got your the DNA. Okay. So the gap around this is around 35 angstroms. And the total diameter of the DNA is around 20 angstrom or 2 nanometer. Like, don't get confused by the number. Okay. In few reference book it is given as 2 nanometer, the total diameter of the DNA, or you can also say that it is 20 angstrom. So inner space is there, okay, for a thin layer of 1 or 2 water molecules also to be present between the sliding clamp and the DNA. Now again, the water molecules are present. Now anyone can tell me why the water molecules are present? Like what will be the importance of having the water molecules between any two molecules? यहाँ पे हो गया आपका sliding clamp and DNA. So why the water molecule is present?
For bonding, ma'am. Hydrogen bonding.
Very good. So every time when water molecule is present, it is because for the interactions. And what kind of interaction a water molecule present can give, that is always the hydrogen bonds. Okay, so it helps in the interactions between the protein. So what is the protein over here? This is a protein, the sliding clamp, and the DNA. So here we have got the small associations or the weak association in the form of the hydrogen bonds. Good. Now, the hole in the center of the clamp is large enough to encircle the DNA double helix and leave room for layer of 2 molecules between the DNA and the protein. Sorry. So, these properties will allow the clamp proteins to sorry, to slide along the DNA without dissociating from it. So it is helping, okay, it is helping to move. So sliding clamp can move along the DNA. So importantly, sliding DNA clamps also bind tightly to DNA polymerase to the primer template junction. Now guys, one thing always remember is whenever you have got a primer template junctions, okay, then only the sliding clamp is present. If you don't have private template junctions, there will be no sliding clamp. And when there is no sliding clamp, then there won't be any DNA polymerase. Okay. So every time when you have a primer template junction, the sliding clamp will be present. Sliding clamp recognizes the association of primer template junction. Okay. And DNA polymerase will be only possible for the DNA polymerase to sit on the DNA helix to synthesize whenever the sliding clamp is present. And overall, when all this sliding clamp, everything is present, then only the DNA polymerase can sit and it becomes easy for it to move along the DNA template to carry out the replication. Okay, guys, is this much clear? The importance of having the sliding clamp? Is this much is clear?
Yes, ma'am.
Okay. Now, how does the association with the sliding clamp change the processivity of the DNA polymerase? Okay, how it is helping? So, in simple, अगर sliding clamp है, the DNA polymerase is going to synthesize efficiently. अगर नहीं है, it cannot. It will fall off from the DNA template. ठीक है? So, in the absence of the sliding clamp, DNA polymerase dissociates and diffuses away from the template DNA while synthesizing around 20 to 100 base pairs synthesize. ठीक है? तो जैसे कि आपके पास है यहाँ पे DNA template है, your DNA polymerase is synthesizing. Okay, but after 20 to 100 nucleotides when it is being added, it falls off from the template. Okay, वो गिर जाता है वहां से. But if you have got sliding clamp, if you have the sliding clamp, what happens is that it is going to keep your DNA polymerase in close proximity of the DNA. It will prevent the polymerase from diffusing away from the DNA template. ठीक है, वो उसको पकड़ के रखता है. It is going to keep together, and that's how it is increasing the processivity or the efficiency of the DNA polymerase in synthesizing DNA. So by keeping the DNA polymerase in close proximity to the DNA, the sliding clamp ensures that the DNA polymerase rapidly rebinds the same primer template junction, increasing the processivity of the DNA polymerase. And this is the second point. First point हमने क्या पढ़ा? Like why the sliding clamp is important. Second point is the importance of having the sliding clamp in keeping or increasing the efficiency of the DNA polymerase. Okay. Yeah, look at the figure now. So now it will be more clear for you. So the sliding DNA clamps, they increase the processivity of the associated DNA polymerase. So as I mentioned before, wherever the primer template junction is there only you will have the sliding clamp. Okay. The sliding clamp, it encircles the DNA and binds to the DNA polymerase. Okay, this is the association. Now, the relatively low processivity of the DNA polymerase leads to frequent release from the primer template junction. अगर यहाँ पे नहीं है तो what will happen? The chances are that the DNA polymerase might fall off from the template. Okay, but since यहाँ पे clamp है और इस clamp ने DNA polymerase को भी पकड़ रखा है, so it will keep engage the DNA polymerase along with the DNA. ठीक है? It will prevent its diffusion away from the DNA. Now, since this is now keeping the DNA polymerase engaged, it is the association of the DNA polymerase with the sliding clamp. It is keeping at the same primer template junction and making sure the DNA polymerase is completing its DNA synthesis. ठीक है? और जैसे ही वहां पे आपका DNA synthesis complete हुआ, like it understands now that there is no other further gap is there, right? Then what happens? Then the DNA polymerase can leave the template. Okay. Now there is no primer template junction is required, right? Because everything has been synthesized. Now the DNA polymerase will leave, and after that, it will the sliding clamp will also get released because there is no further template primer or primer template junction. Okay. Yeah. So guys, this is clear. This is really very important to understand the replication. Okay, everyone, for everyone it is clear?
Yes, ma'am.
Yeah, okay.
Yes, ma'am.
Tanvi, what about you?
Yes, ma'am.
Okay, so let's move further then. Now, sliding clamps, they are opened and placed on the DNA by the clamp loader. ठीक है? अब यहाँ पे क्या हो गया? ये हो गया आपका दूसरा प्रोटीन. What we are talking about is clamp loader. So, अभी हमने देखा sliding clamp क्या है? It's again a ring-like structure which opens up at the DNA and उसके ऊपर ही आपका DNA polymerase भी बैठता है. ठीक है? So it's a closed ring in solution, but must open to encircle the DNA double helix. It's a special class of protein complexes called clamp loader. They catalyze the opening and placement of the sliding clamps on the DNA. If you remember in the previous figure, हमारे पास एक था main protein, like a very bulky protein. It had the claws like kind of structures. So those are the top proteins, and उसको hold कर रहे हैं वो हो गया आपके clamp, sliding clamp. जो उसको बना के रखा है वो आपका clamp loader हो गया, ठीक है? Maybe for example in this I can explain. So this is your clamp loader, okay? Clamp loader है यहाँ पे. Now what you see is with the help of the ATP, see, can you see it over here? With the help of the ATP, it is opening the sliding clamp. Okay, once it is open, along the DNA, it is wrapping. And जैसे वो clamp में लग जाएगा, जैसे clamp is able to load on the DNA, the loader will leave. Okay, and यहाँ पे now the DNA polymerase can come and synthesize till the gap is present. Okay. I'll show you another figure in that it will be more clear. Just have patience. So these enzymes, they couple ATP binding and hydrolysis to the placement of the sliding clamp around the primer template junctions. Okay. So clamp loader, they also remove sliding clamps in the DNA when they are no longer in use. And it does not require ATP hydrolysis. किसी भी प्रोटीन का conformational change लाने के लिए you always need ATP hydrolysis. ठीक है? इतना by default याद रखो. Okay, yeah, this is a very good structure. The figure. Again, this figure has been taken from the Watson only. Okay, so look at over here. So you have got the sliding clamp loader over here. It's a 5 subunit protein and all. Okay, now, of course, in the eukaryote cell, it is known as replication factor C or RFC. Same thing, okay? Now, you need to keep this clamp open. To open this clamp, you need ATP. And you can see that where the ATP binding is happening. So ATP is there. ATP goes and sits at the binding pocket of the clamp loader. This is your clamp loader. Okay. ATP is sitting over there. Now your sliding clamp is coming. With the help of the ATP, it is able to open the sliding clamp. Okay. ये loader हो गया, ये sliding clamp हो गया. Now once it is open, the clamp loader wraps around the DNA sliding clamp, okay, and then the ATP hydrolysis happens and gets dissociated. इतने में क्या हुआ? आपका sliding clamp जो है DNA के around wrap हो गया है. Is the figure clear to everyone? What exactly is happening? I have got the video also ready. Okay, you can watch in that. But before that, we need to clear these topics. Yes. Is everything clear till this much?
Yes, ma'am.
Okay. Now, DNA synthesis at the replication fork. So we know how exactly the DNA is getting synthesized at the replication fork. हमें मालूम है. But now you need to talk about all the proteins together. अभी हमें कैसे पता है? Like only DNA polymerase acting at the replication fork, and आगे आगे helicase चल रहा है, पीछे पीछे you have got the single stranded binding protein and then DNA polymerase coming and synthesizing the DNA, right? Now we need to include the concept of clamp loader as well as sliding clamp. Okay. So at the replication fork, the leading and the lagging strands are synthesized simultaneously. Okay, and limiting the amount of single-stranded DNA present in the cell during DNA replication. Now this is also very important because you can't keep DNA single-stranded for a long time because this will be considered as a mutation in the cell. ठीक है? तो जैसे ही आपका डबल हेलिकल सिंगल स्ट्रैंडेड हुआ ऐसे, ओके, यहाँ पे आपका हेलिकेज आगे आगे जा रहा है एंड यहाँ पे आपका डीएनए पॉलीमरेज सिंथेसाइज कर रहा है. तो as soon as the DNA strands are getting single stranded, very fast, very rapidly, the DNA polymerase are synthesizing and making the new strands of DNA. Okay. Okay. So when a single stranded DNA is broken, there's a complete break in the chromosome, and then that should undergo repair. अगर नहीं होगा repair, what will happen? That particular cell will undergo apoptosis. If you cannot repair the cell, then the entire cell will go into the apoptosis. Okay, yeah. Now, so moreover, repair of this type of lesion frequently leads to mutation in the DNA, and that's why having the limiting time where the single-stranded DNA is present is crucial. So to coordinate the replication of both strands, multiple DNA polymerase function at the replication form. Now we'll see that एक बार में कितने DNA polymerase हमें लगते हैं, ठीक है? Now look at the figure. Look at the figure. So this is your sliding clamp which we were talking about. अब ये open structure है, ठीक है? ये हो गया आपका clamp loader. Now this clamp loader is holding your DNA polymerase. Now how many DNA polymerase are present? There are 3 DNA polymerase present. Right? Now the question is why 3 DNA polymerase जबकि हमें 2 are required for DNA replication. Now who can tell me why the third protein is present, third DNA polymerase is present? Yes, Akshaya, what do you think? Why we have got 3 DNA polymerase instead of 2?
maybe for stabilizing or for any repair kind of.
No. When replication is going on, there is no repair happening. If repair is happening, then no replication is happening. Yes. What about Chandni, you? What guesses can you have?
No idea, ma'am.
Okay. Shruti? Tanvi?
There is leading and lagging strand 2 DNA polymerases. Maybe I'm just guessing.
Okay, but that's what I'm saying. Na? 2 are required for leading and lagging. Third की जरूरत क्या है?
Yeah.
Okay, it's okay. No problem. I'll tell.
Ma'am,
Yes.
because it can able to bind three polymerases, ma'am. The
No.
loader have 3.
Loader can have 3,
proteins
loader
like
can have 4 also. But at a time, it is bound with the 3 DNA polymerase. See, when there is a requirement, then only the protein is going to do the function, right? If there is no requirement, no need to do the function because it involves a lot of amount of energy. Okay, we don't simply spend money on the stuff we don't want, right? In the same way, cell don't spend their energy on the stuff which they don't require. So here it is important to have yes.
Is it because is it DNA Paul the repairing the ones that repairs the Okazaki fragments, that one?
Nahi. All the 3 are DNA fall 3. Okazaki fragments gap filling is fall 1.
Yeah, that's why. Since only one was labeled as Paul 3, so I thought maybe
No.
one of them was Paul 1.
No, all the 3 are same and DNA Paul 1 is freely present in the nucleus. It is not bound to anything. Remember, I have always said the DNA Paul 3 is the hollow enzyme. Now, when you say hollow enzyme, it means the complete enzyme along with the tau proteins, sliding clamp loader and the sliding clamp. This entire thing is called the hollow enzyme. Only this particular portion is called DNA Paul 3. If total is the hollow enzyme. Yes. Yes, anyone was saying something? Okay, so no problem. See, if you remember in the case of the lagging strand, I have mentioned that the loader can move only in one direction, right? This direction में आपका leading strand जा रहा है. And in the same direction, your lagging strand is also going. Only difference is that it just brings out the loop-like structure so that the DNA polymerase can synthesize in this direction, right? अभी क्या होगा? The moment it is synthesizing this much portion, आगे तो आपका ये free हो गया है, right? New primer has been set. So by the time this is synthesizing and take free, this region will be taken in front and second polymerase इसको bind करेगा. So 1 and 2 is always associated with the lagging strand and the 1 is fixed for the leading strand. Okay. Okay, maybe the diagram is not clear, but the concept is clear. I'll show the video, then I guess it will be clear to you guys. Okay, itna yaad rakho that the two are associated with the lagging and one is associated with the leading strand. Okay, itna yaad rakho. I'll show the figure as well as the video. So how do the multiple polymerases are linked at the replication fork while synthesizing DNA on both the leading and the lagging template strands? कैसे होता है? तो एक model भी propose किया है and that came out to be true. Okay, so many people have made the animations and to get the clear idea about it. Okay. So this T is tau protein, okay? So as the helicase unwinds the DNA at the replication fork, the leading strand template is exposed and acted immediately by the DNA Pol 3 core enzyme, okay, which is going to synthesize continuous strand of the complementary DNA. Whereas the lagging strand is not immediately acted on by the DNA polymerase. It takes time. Now, why it is taking time? Because it needs to bring the loop ahead in front of the DNA polymerase. Then only it can start synthesizing. Okay, now look at the figure. Okay, so which direction it is going? Can you tell me? Yes, anyone? See, यहाँ पे आपका DNA helicase है. This is your double-stranded DNA, right? We'll go slowly, okay? Here you have got your helicase. So double-stranded DNA, helicase, replication fork is in this direction. Look at the leading strand DNA polymerase. So you have got the sliding clamp, tau protein, and 3 associated tau proteins, which is holding together your DNA polymerase. Okay. Now this is clear. Like DNA polymerase is there, sliding clamp is there, and it is associated with the synthesizing on this strand. So ये portion clear है आपको? Yes, Akshay agree?
Yes, ma'am.
Now
Yes,
second. Now look at the second figure. Not second figure, the second part. So as I said, अभी ये direction में we need to go into this direction. Replication fork can go in one direction at the same time, right? तो वो कौन से direction में जाएगा आपका leading strand के direction में जाएगा, right? But the problem is that I need to synthesize lagging strand in this direction क्योंकि इस direction में इसका 5 prime 3 prime आ रहा है, correct? इस direction में जाना है. So what does the holoenzyme does is it will wait, okay? It will wait for this much region to be open up. Then what it is going to do? See, ये आपके SSB proteins है, single stranded binding proteins are there. Okay, जैसे ही वो आगे आएगा, it will come in front of this, the DNA polymerase. तो वो उसके फ्रंट में आएगा, वो यहाँ से प्राइमर रेडी होके उसने वहां पे सिंथेसाइज करना शुरू कर दिया. ठीक है? See, either you can take loop ऐसे भी ले सकते हो और आप loop ऐसे भी ले सकते हो. But it is going in this direction. आ रहा है क्या? क्लियर हो रहा है थोड़ा बहुत?
Yes, ma'am.
Now what happens is जैसे ही see it has synthesized and reached this much area. ये SSB हटा देगा. It is reaching this area. So what is happening now? There are no further primer template junction. So this DNA polymerase will leave. Okay, but यहाँ पे तब तक ready हो गया आपका single stranded DNA along with the primer ऐसे. So by the time the second DNA polymerase will come and attach and start synthesizing. वो जैसे यहाँ reach करेगा, it will leave. जैसे ही second करेगा तब तक उसके third loader में, third term protein में, third DNA polymerase will be ready to be loaded. along with the sliding clamp. Okay. Abhi, I'll do one thing. I'll show you the figure, rest of the figure. So this is how it is going to happen. ठीक है, मैं आपको एक बार वीडियो दिखा देती हूँ. Maybe that time it will be clear. Watch the video carefully, okay? We'll come back again. So this one is your leading strands, okay? जो नहीं छोड़ रहा है the DNA polymerase. यहाँ आपका हेलिकेज है आगे की तरफ जो ऐसे जा रहा है. Can you see this light green color is the sliding clamp. So leading strand में आपका constant है. अब यहाँ पे देखो, जैसे उसने यहाँ पे इतना synthesize किया, this will leave, and by the time the second is getting ready to be loaded. Okay. हाँ, अभी थोड़ा सा आइडिया आया.
Yes, ma'am.
थोड़ा बहुत. We'll see the video again. So what you saw over there was every time on the leading strand the DNA polymerase is constant. ये particular portion जो है आपका constant है, ठीक है? ये बार बार change नहीं होगा. This is just going smoothly synthesizing. Whereas this, the loop is coming and the DNA polymerase is able to synthesize and leave. By the time second sliding clamp along with the DNA polymerase is getting ready, वो यहाँ पे आके बैठेगा. फिर ये यहाँ तक ऐसे synthesize कर आएगा. It will leave. Then third, then fourth, ऐसे ready होगा. तो इसलिए हमें two top rotates are required for the lagging strand. ठीक है? We'll go now step wise. So, okay, so the resulting RNA, okay, this is not required. So this model, let's talk about how the DNA replication is happening, and it is known as the trombone model. Okay. Yes.
Okay. Yeah. So just for convenience sake, I'm going to say that DNA pol 1, 2, 3, and are all the DNA polymerase 3, and just the numbers. I just want to refer to them.
Okay.
So there's one of the DNA pols which just goes along the leading strand and just continues until it runs out. And then there's another DNA pol that actually starts after leaving some part, which is a part of the loop, so that the direction is used. The direction is the same as the first DNA pol, and that continues on until it drops off. And by that time, the part which was left over is ready to be replicated, which is taken over by the third DNA pol. That is what happens, right?
Yes, yes. See, कैसा होगा जब तक ये इसको synthesize कर रहा है, the helicase is separating the strand. Leading ने तो अपना synthesize कर लिया, right? But the DNA polymerase is not available over here to synthesize. So तब तक के लिए single stranded binding proteins ने उसको bind कर रखा है, okay? And by the time it is reaching, अब क्या होगा, ये यहाँ से छोड़ देगा, then the second will come and start over here. By the time ये ऐसे synthesize करते 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 यहाँ पर आएगा, again by that time the another area, the loop is free for it to bind. ठीक है? In the video also the same thing is happening. I'll show slowly also कैसे हो रहा है, ठीक है? So let's study first all the steps, how exactly it is happening, and then I'll show the video again. Okay? Okay. I'm waiting for Chandni to join. Okay. Yes. So now everyone is back. Yeah. So the DNA polymerase, DNA pol 3 holoenzyme, it interacts with the DNA helicase through the tau subunits, which also bind to the DNA polymerase 3 core proteins. Now this is also important. Okay. The holoenzyme is interacting with the helicase. तो जैसे जैसे आपका हेलिकेस आगे बढ़ रहा है उसी स्पीड से आपका holoenzyme is also moving. ठीक है. Now, one DNA pol 3 core is replicating the leading strand while the other two are dedicated for the lagging strand. Okay. Now, sorry. Now, DNA primase associates with the DNA helicase and synthesizes new RNA primer of the lagging strand. Now, एक चीज और याद रखनी है that primase is getting activated by the activity of the helicase. Now, in the video, थोड़ा सा ध्यान देना. To the helicase, something comes and bind. Okay, that something is nothing but the primase, which gets activated and then only it is able to bind. तो आप ध्यान देना वहां पे प्राइमेज आएगा. Now can you see it? This is leading strand. Okay, so leading strand आप छोड़ सकते हो just to understand how lagging strand में हो रहा है हम लोग वो भी डिस्कस कर रहे हैं. ठीक है. So you can see over here the primase comes and gets activated and primer बना देता है. ठीक है. Now you can see that यहाँ पे तब तक आपका primer ready हो गया है. Correct. But this DNA pol 3 has synthesized this much and it will synthesize still here and then this DNA pol will follow. Okay, अभी primer यहाँ पे ready है. So by that time your this third one, ये second हो गया, ये first हो गया. The third DNA pol 3 is ready now to get loaded over here. जिसका sliding clamp यहाँ पे रखा है, ready, open करके ready रखा है. तो जैसे ही ये आगे बढ़ेगा, primer ने अपना काम कर दिया. Right? So what is happening now? As I said, sliding clamp के लिए क्या जरूरी है? To have primer template junction. Right? So जैसे ही यहाँ पे your primer template junction is ready, the clamp loader is going to put the clamp around over here. और जैसे ही clamp ready हो जाएगा, DNA polymerase the third one है, it will come and sit over here and will start synthesizing. ये तब तक आपका गिर जाएगा. ठीक है. By that time आपका ये third one हो रहा है. Again the clamp is ready and the DNA pol to the third protein is ready. Okay. So this is how during the elongation the DNA polymerase is synthesizing. Is this much part is clear?
Yes, ma'am.
Huh? Okay. Now immediately after the new RNA primer is synthesized, sliding clamp loader, sorry, the loader is assembling the DNA clamp at the resulting primer template junctions. As I said, primase हट गया अभी. The moment it is removed, what is happening? The loader has put the clamp over here, right? So once the clamp is put over here, this DNA polymerase will come and sit over here and will start synthesizing this single-stranded DNA. अभी हमारे पास primer है, DNA polymerase है, and DNTPs are present in the pool, so it can take it from there. Look over here. तब तक आपका clamp भी आ गया, अभी DNA pol भी आ गया. Now look at over here. This is almost going to complete, right? So after that, this is going to leave, and by this time, this is ready. तब तक ये यहाँ पे synthesize करेगा, and you can see at the back, again the single-stranded DNA is free. ठीक है? So for this portion is going to get ready. Again फिर से यहाँ पे sliding clamp आएगा, DNA pol आएगा, and it will get ready over here. Is this much clear now? Yes.
Yes.
Now this has started synthesizing and यहाँ पे आपने देखा, see it has left. Okay, now it has left. Eventually this will also remove. Now जब तक यहाँ पे second primer नहीं आता और उस पे आपका sliding clamp नहीं आता, the third DNA polymerase cannot sit. जो आपका second था अभी वो आपका third हो जाएगा. So the position of second and third polymerase will be changing. जो 1 है दैट इज मेंट फॉर लीडिंग, द सेकंड एंड थर्ड इज मेंट फॉर लैगिंग। तो ये आपका बारी बारी करता रहेगा। ओके, so this is the proper model of DNA replication. So same thing is written what we have discussed. So as one lagging strand DNA polymerase synthesized in Okazaki fragment, additional single stranded DNA is generated by the helicase and तब तक आपका new primer भी ready हो जाता है। Okay, now as the lagging strand primer, the new primer is recognized by the sliding clamp loader. और जैसे ये लोड होगा, लोडर आइडेंटिफाई करेगा, इट इज गोइंग टू पुट द न्यू स्लाइडिंग डीएनए क्लैंप, एंड जैसे ही क्लैंप रेडी हो जाएगा, आपका डीएनए पॉलीमरेज भी आ जाएगा। Okay, so as your each Okazaki fragment is completed, the responsible DNA polymerase is released from the template and new it will come. So because the release of the DNA polymerase from the sliding clamp is slower process than the DNA synthesis, having a second DNA polymerase dedicated for lagging strand synthesis is ensuring that lagging strand synthesis is also continuous even though it's a slow process, slow polymerase release. ठीक है, नहीं तो क्या होगा, just imagine suppose only one DNA polymerase was dedicated, it will wait and by the time the loop जो इतना बड़ा जो हमारा फॉर्म हो रहा है वो बहुत कलेक्ट हो जाएगा, तो there are chances that the single stranded DNA might get tangled up to each other and might break. ठीक है, तो हमें हमारा रेप्लिकेशन का प्रोसेसिविटी फास्ट रखने के लिए we are making sure that we are using 2 DNA polymerase specially dedicated for the lagging strand. Okay, now look at the video now. ध्यान से देखना, मैं बीच बीच में रोकती भी रहूंगी टू एक्सप्लेन। ओके, कैन यू सी इट ओवर हियर? आपको टॉ प्रोटीन समझ में आ रहा है? दिस इज योर लोडर, कैन यू सी दीज हैंड काइंड ऑफ थिंग? ये आपके टॉ प्रोटीन है, ठीक है। दिस इज योर क्लैंप, स्लाइडिंग क्लैंप। देखा, वहां पे आके स्लाइडिंग क्लैंप बैठा, द मोमेंट इट सिट्स ओवर देयर, ये क्या गया आपका प्राइमेज। ओके, इट इंटरैक्ट्स विद द हेलिकेज, गेट्स एक्टिवेटेड, प्राइमेज वहां पे प्राइमर बना देता है, देन योर क्लैंप कम्स सिट्स ऑन द लोडर, लोडर इज गोइंग टू पुट द क्लैंप अराउंड द डीएनए। सी अगेन द प्राइमेज हैज कम, वहां पे उसने येलो कलर का प्राइमर बना दिया। दिस इज द स्लाइडिंग क्लैंप, उसे सीधा बनाया, जैसे ही यहाँ पे खत्म हुआ इट लीव्स। See, this one is your primase, उसने प्राइमर बनाया येलो कलर का। Yes, everyone, are you able to follow?
Yes, ma'am.
This is your helicase, okay? यहाँ पे आपका डबल स्ट्रैंडेड डीएनए है, हेलिकेज है, इट इज ओपनिंग। तो यू कैन सी दैट ये लीडिंग स्ट्रैंड है, ये अपना स्मूथली चल रहा है। प्रॉब्लम कहाँ की? लैगिंग स्ट्रैंड में। यू कैन सी यहाँ पे सिंगल स्ट्रैंडेड डीएनए है, एक्सपोज्ड बेसिस है यहाँ पे। ओके, सी यहाँ पे क्लैंप आपका रेडी हो गया है, यहाँ पे सॉरी, जैसे ही आपका डबल स्ट्रैंडेड बन गया, यहाँ पे देयर इज नो फर्दर प्राइमर टेम्पलेट जंक्शन, सो डीएनए पॉलीमरेज हैज लेफ्ट, only clamp is present. So what will happen now? The loader has left this region, so sliding clamp हट जाएगा, but by that time it has kept ready over here. सेकंड से रेडी रखा था, अभी डीएनए पॉलीमरेज यहाँ पे देखो, ये पर्पल कलर का डीएनए पॉलीमरेज है, इट इज सिंथेसाइजिंग। लेट्स वॉच अगेन। अभी दोनों ही केसेस में क्या होगा, एवरीथिंग इज मूविंग इन दिस डायरेक्शन, लीडिंग ऑल्सो, लैगिंग ऑल्सो इन दिस डायरेक्शन. Okay. Now let's talk about the regulation of DNA replication. Now, what kind of regulation we are talking about over here? Okay, before this, like I should ask, is it clear with this replication is over? Okay, with that video in the prokaryotes, and there will be another short session for the eukaryotic. कुछ ज़्यादा अंतर नहीं आएगा because you just need to remember the different names of the proteins associated. जैसे कि, for example, in the case of prokaryotes, you have got the ORIC, right? In the case of eukaryotes, you are going to have ORC, origin recognition complex, and the other proteins associated with that. Okay, much difference is not going to come. Instead of DNA polymerase 3, there we have got the DNA poly alpha, beta, gamma, delta. इतना ही difference आएगा. So anyways, so before we shift to eukaryotes, are you guys, satisfied? Like, do you understand DNA replication now? Yes. Okay, not 100%, but once you refer it, do you understand now कि हाँ इस तरह से हमारे बॉडी में या प्रोकैरियोट्स में डीएनए रेप्लिकेशन होता है. The concept is clear to you. Later you can remember the names of the proteins and all. That is different story. But do you understand the concept behind the DNA replication? कैसे लैगिंग को भी साथ में लीडिंग लेके चल रहा है? Okay. Okay. So that is important for me. So now let's talk about the regulation. Now, when we talk about regulation, what kind of regulation we are talking here? It is that in one time, in one cell, the replication should happen only once. Okay. Now, how it is achieved? That is what we are going to discuss. So, in all organisms, it is critical that replication initiation be tightly controlled to ensure that chromosome number and the cell number remain appropriately balanced. ठीक है? नहीं तो क्या होगा? If the number changes, you know that aneuploidy. Even if you have more number of chromosomes or less number of chromosomes, it is going to result into the genetic disorder, right? So that's why it is important that in the cell, one time, okay, the replication should happen, and then only the division should happen. Now let's talk about in the case of E. coli. So we know something about the DAM methyl transferase. So earlier we talked about the GATC sequences also. Okay. So we know that the DAM methylase, they add the methyl group at the adenine. कहाँ पे? जहाँ पे भी आपका GATC sequences है. Now, what can you tell about these sequences? This is your ORIC. Remember, 13 mer and 9 mer units are there, 5, 9 are there, and 3, 13 are there, right? So this is nothing but the ORIC. Now, what do we see over here? Before replication, GATC, जहाँ पे भी आपका GATC है, it is methylated. Okay. Can you see? Can you understand? So this is before replication. So you have got the methyl group present everywhere, wherever you have got the GATC sequences. So the genome is fully methylated. Now this situation is changed once after each GATC sequence is replicated. So now what will happen? The daughter strand is not going to have the methylation, correct? The daughter strand is not going to have the methylation. Only the parental strand is going to have the methylation. And the daughter strand is not having. Parental strand is having. So that condition is known as hemimethylation. Right? So this hemimethylated state of newly replicated ORIC DNA is detected by a protein called SEC-A. Okay, SEC-A. Now, this is going to bind, this SEC-A is going to bind tightly to the GATC sequence only when it is hemimethylated. Okay, now heavy methylated कब होगा? जब आपका replication हो जाएगा. So parietal strand and daughter strand is there. Parietal having the methylation, daughter strand not having the methylation. Okay, and we know that near ORIC there are so many GATC sequences. So once replication has initiated, sec-Q binds to these sites before they can become fully methylated by the DAM methyl transferase. Guys, is this slide clear to you? So what we have discussed? We have discussed that after the replication, you have got the parental strand methylated, right? And daughter strand not methylated. So this condition is known as heavy methylated, and to this heavy methylated condition, the protein is sec-A protein comes and bind. Okay, and we know that at ORIC there are many GATC sequences. Okay. Now, the significance of binding of sec-A to the heavy methylated ORIC. So what is happening is that if you remember, in the case of ORIC, we have got those 5 units of 9-mer sequences, correct? And DNA binding protein कहाँ bind करता है? It binds to these regions only. Okay, so now at this hemimethylated region, when sec-A protein goes in bind, it is blocking, it is preventing the rebinding of the DNA A binding protein. Okay, so it is preventing the new round of replication. अगर वहां पे sec-A protein नहीं होता, then DNA A binding protein will go again and bind to these sequences and will reinitiate the replication. But we have got the sec-A proteins to inhibit or to prevent the second round of replication. Okay, so this is going to prevent the rapid replication while the first one is going on. Is this much clear to everyone? Yeah.
Yes, ma'am.
Yes, ma'am.
Get the figure, it will be more clear now. Okay, so you have got the ORIC over here. Okay, this is your ORIC. Now you can see that both the strands are methylated, right? Now initiation of the replication. So you can see that the daughter strands is not methylated. The moment this region, the ORIC region is getting replicated, the sec-A protein will come and bind wherever you have got the GATC sequences, and only hemimethylated state it can bind. Always remember. ठीक है? अभी just imagine अगर ये sec-Q protein नहीं होता, so what will happen? ये sequence यहाँ भी present है, ये sequence आपके newly daughter strand में भी present है, right? So the DNA A binding protein can come and start the another round of replication. यहाँ पे भी शुरू हो जाएगा, फिर यहाँ पे भी शुरू हो जाएगा. Okay, before the previous replication has been getting completed. So इसलिए sec-A protein will come and bind over here. Okay. So now what is happening? Now the DAM methylase cannot methylate. DNA binding protein cannot bind. Okay. So it is preventing. Now once prevention has been done, okay, sec-A will leave. Once sec-A is leaving, then dimethylase can come and methylate the daughter strand. Okay. Now once it is methylated, now the DNA A protein can come back again and start the reinitiation of the replication. Clear? The regulation of DNA replication, is it clear? Now, this is also important because many a time question has come. And this is only possible in the case of E. coli. So, there is always a question they ask that we know that E. coli at optimum conditions like good temperature and 37 degrees Celsius takes 20 minutes to divide. Okay. But the entire replication of its genome takes 40 minutes. So कायदे से देखा जाए, it should take another 20 minutes to divide, but we know that E. coli takes only 20 minutes. उसका generation time 20 minutes है to divide, but उसका एक जीनोम को पूरा replicate होने में 40 minutes लगता है. So how is it possible? So in the case of E. coli, what they do is, so in the case of E. coli, what happens is that, as you can see in the figure, so कैसा दिख रहा है? You have got the E. coli inside the genome is there, and you can see that the moment it is reaching its termination sequences, the second round of replication has started. है ना? So while it is getting separated, cell division जब उसका हो रहा है, along with the cell division, you can see that the second round of replication has already initiated. Yes? Are you able to follow?
Yes, ma'am.
Yeah, and that's why we say that it takes only 20 minutes for the DNA, for the E. coli to divide, but the entire time for the replication is taken is 40 minutes. और जैसे ही इसका second round होगा, जैसे ही it is reaching its replication end over here, termination, what will happen? Another bubble you can see over here. It means for the next round it is getting ready. Okay. So being actively replicated, the cell gets divided. But this is not true in the case of the eukaryotes. Eukaryotes में जब तक सब कुछ नहीं हो जाता, it will not undergo M phase or the mitosis. Okay, ऐसा नहीं कि आधा divide हो गया and then, because see in the case of eukaryotes we don't have circular DNA, right? We have got 23 pairs. जब तक वो अच्छे से align नहीं होते एक दूसरे के साथ, तब तक उनको हम डिवाइड नहीं कर सकते. So that's why in the case of eukaryotes it is not possible. Okay, is this much clear to everyone? Yeah. So now I'll be having MCQs for you.